Okay, so we have a lot to go through today. So one of the things that I have added to help you guys out, and I am going to get rid of an objective. I'm sure that'll make you guys happy. It's a kind of a big one. So under the PowerPoints, as I said, I added one small one. It's only 6 slides. I clicked on the wrong one. Wrong module. We are in 2. I'm going to get there. Yeah, it's small. Okay. Okay. So. And this was towards the end of class, and so most people weren't listening to me anymore. Right? Now it's taking a minute to connect for some God only knows the reason. Okay. So, I even put like the date. I updated that too to help you guys out. But this little PowerPoint right here is like 6 slides. It covers this one last objective for growth and control, which the topic biofilm is talked about in Chapter 15, Section 1, in quorum sensing, and Chapter 11, Section 5. This objective, it said refer to a table, which of course you guys don't have. That was from the old book. So I actually wrote out what was in the table, basically all these terms that you need to be able to define. The objective that I am going to post an announcement here and take out is also related to one of the tables and stuff. And so I had also had, you know, all the tables that would help you study. I put in a separate PowerPoint for you guys and diagrams. I think I already have this one open now. All right, so I meant for this one to be before this one so you could like, yeah, fill it out and then check your answers. But so it doesn't have the pop-ups, right? But this one, I think I even didn't put it in here. I didn't even put it in here. Haha. Good. I don't have to take it out. It's not even in there. But it's in my PowerPoint. So we'll get to that. So, let's see. Where were we? We talked about the different nutritional categories. You definitely need to know those and be able to work through them. So then I started to want to talk about oxygen requirements. So you'll notice on this one, as I said, oxygen concentration, right? We need to know how this influences growth. And you need to know these terms as it relates to oxygen tolerances. So I have a couple of tables and diagrams for this one to help you out. And what I have today too, see if I can, there we go. I can pull it up, yay. All right. Is some show and tell. So let's see if you guys can see this. I need a white background. I got to go the other way. Find it, Erica. It's opposite. So I'm having a dyslexic moment. Do you guys see how there's a pink area in this tube? At the top, right? So this is one of those FTM tubes that we use to determine oxygen tolerances in the microbiology lab. So that pink layer is an indicator that lets us know that there's a high concentration of oxygen in that area, where the other area where you don't see the pink at the bottom of the tube, there isn't any oxygen. So I have these up front. These are glass tubes. I'm not going to pass them around. But when class is over, if you want to come up and see it up close, feel free to do so, and I'll let you know, let you see it. So I have a couple of other ones where I'm going to pop this up and show it to you guys. So let me go to, can I move this? Yes. Hopefully it's not completely gone. Okay. So you need to be able to define those 5 categories, right, for oxygen tolerances. You need to be able to identify the categories that actually use oxygen. This one's pretty easy as we walk through this. It'll make sense to you. You need to be able to name the enzymes that help protect organisms against the toxic forms of oxygen. That's why we refer to as oxygen tolerances even. And then you also need to be able to identify those growth patterns in FTM tubes, of course, in a drawing as opposed to the actual tube. In lab, we use the actual tubes. And then we're going to talk about the problems that you run into in trying to grow anaerobes, right? And what are some of the tools and stuff we use at home? Because remember, one of the things is we talk about growth, then we can also flip this and say, well, how can we use this information we know about growth to therefore control the growth of organisms? So if an organism needs oxygen to grow, how could we slow or stop its growth? Remove the oxygen. So there are some fancy machines and even not so fancy pumps you can buy nowadays that you can literally suck the oxygen, the air, out of the container in which you're storing food in, right? I don't want to advertise for any of them, but you know, the most common one is like Food Saver, right? So, okay. So, part of what we see has to do with the fact that oxygen can be toxic, right? So for some organisms, even the ones that live in the presence of it, they've got to deal with this problem. So you can see there's several different forms of oxygen that is toxic. And some of them we produce even. As we're utilizing oxygen, we produce these toxic forms. It's a byproduct. It's an unfortunate byproduct. So we have defenses against that, or at least some organisms do, and that's why they can live in the presence of oxygen. And in our case, we can actually utilize oxygen. So we're going to talk about these, one of them being superoxide demutase. So as the name implies, it helps deal with these superoxide anions that can form. These negatively charged oxygens. They have more electrons than they're supposed to. Makes them dangerous. The other is catalase, and that one helps detoxify hydrogen peroxide, which is something you might even use at home, right, as a disinfectant or antiseptic, although they suggest that you don't use it anymore. It's actually not recommended that you use it for wounds anymore. Soap and water and good old triple antibiotic ointment. We're going to talk about it. Okay, so let's first go over our definitions of each one of the classifications. So aerobes, referring to air, right, aerobic, they require, absolutely have to have oxygen. So it's a lot of times they're referred to as obligate or even strict aerobes. And that's us, right? We can't grow without oxygen. We cannot live without oxygen. We will die. There's a subset of aerobes referred to as microaerophiles. So as the name implies, right, micro means what? Small. These are organisms that require small concentrations of oxygen. Now think about this. We live in oxygen. We use oxygen. So again, we're producing toxic forms of oxygen and we're exposed to toxic forms of oxygen by living in an oxygen-rich environment. So what do we have to protect us? The enzymes, right? Superdeoxide mutase and catalase to help break down those toxic forms and make it safe again. So if we're a microaerophile, we require only small amounts. So in other words, just enough to do our metabolism, right? If you're a microaerophile. But why can't we handle more? What do you think? Why can't a microaerophile just live in an oxygen environment and just use less oxygen? Why does it actually have to live in an environment where there is less oxygen than normal atmospheric conditions like what we're living in? What would happen to them if they were at atmospheric? What's the problem about living in an oxygen environment? It's toxic, right? So you have to have protection. So do you think they have the same level of protection as we do? They have less protection. Therefore, they can only deal with lower amounts of oxygen. But they still need it. They've got to have just enough to be able to metabolize. Does that make sense to you guys? So the next one is aerobe. So now we're talking about without oxygen. So these are organisms that have the ability to metabolize without oxygen. So anaerobe meaning without. This does not mean they don't use it. It means they have the ability to not use it. Make sense? So there's 3 classifications here. Aerotolerant anaerobes. As the name implies, they can tolerate what? Aerotolerant. What are they tolerating? Oxygen, right? Because oxygen is what? Toxic. So guess what? They have a good level of protection. But the fact that we're calling them an anaerobe, are they actually using oxygen for their metabolism? No, right? We call them an anaerobe because they're not using oxygen for their metabolism. We call them aerotolerant because they can live in the presence of oxygen. They do have protection. It won't kill them. Obligate or strict anaerobes, on the other hand, it's an anaerobe, so is it using oxygen? If it's an anaerobe, is it using oxygen? No. And in this case, cannot grow when oxygen present. Why not? Oxygen is what? Toxic. So if it's toxic and you don't have what? Protection, you don't have those enzymes, it's going to kill you. And that's what happens to anaerobes, right? If you put them in an oxygen environment, you're going to kill them. Facultative anaerobes are probably the best suited for the world. Facultative means the ability to change. Anaerobe means they can do what? What does anaerobe mean? What can they do? They can grow without oxygen. So these guys can grow without oxygen, but they do have the ability to grow in the presence of oxygen. So facultative, they can change, right? So oxygen's available, they'll grow really fast because oxygen is the best as far as energy yield. They'll grow super fast. But when oxygen isn't available, instead of dying, like most aerobes like ourselves, they can switch to an anaerobic form of metabolism. Most commonly, they ferment. Right? So remember how we talked about nutritional categories before and how we're stuck being chemo-organoheterotrophs and plants are stuck being photolithoautotrophs, right? There are some organisms that can change, right? They have the machinery to do both, depending on what's available, right? So a facultative anaerobe, as I said, is probably the best, right? Because he's like, I can do whatever they throw at me. I'll be fine, right? I can survive oxygen, I can use it, I can grow fast. Or if it's not there anymore, I'll just ferment. No problem. Yeah, most of the organisms that are in your gut, like E. coli, are facultative anaerobes. Yeah, they can adjust. There's lots of facultative anaerobes out there. Because again, that's the best choice, right? Because you can do it all. You can do it all. So let's see if we can fill in this table based on the definitions we just learned. So, obligate or strict anaerobes, do they require oxygen for their metabolism? Do we have to give it to them? Yes. Can they grow in the presence of oxygen? Yes, and we can almost add to this question, can they tolerate oxygen, right? Do they have the enzymes to protect them against oxygen? And the answer is yes, right? Otherwise, they'd be in big trouble because they need it, right? They have to have it for their metabolism. Can they grow without it? No. They don't have anaerobe in their name? Answer's no. Obligate strict anaerobes. Do they require oxygen for their metabolism? Definitely not. Can they grow in the presence of it? No, right? They don't have any protection. It will kill them. Can they grow without it? Yeah, as their name implies, they're an anaerobe. They can do that. Microaerophiles. Do they require oxygen for their metabolism? Yes, but in small quantities, right? Can they grow in presence of oxygen? Yes, but there's another but here, right? Small quantities because their protection is limited. Can they grow without it? without oxygen? No, right? We've already said they require it, so they certainly can't grow without it. Do facultative anaerobes require oxygen for their metabolism? No, right? They don't require it. They're an anaerobe, which means they don't require it. But can they grow in the presence of it? Yes, they can, right? They have protection, and they will, in fact, grow very abundantly because oxygen gives you the best yield for energy. Can they grow anaerobically? Yeah, there's anaerobe in their name. Yeah, they'll grow at a modest level. You don't get as much energy when you don't use oxygen. But hey, it's better than dying, right? I'd be like, okay. Aerotolerant anaerobes. Do they require oxygen for their metabolism? No, because they're an anaerobe, right? So they don't require it. Can they grow in the presence of it? Yes. They do have protection, as their name implies, aerotolerant. But it doesn't affect their growth. And you'll see this in a moment. It doesn't affect their growth because they're not using it, right? Unlike facultative anaerobes, they do actually use it, so it does affect their growth. They don't have to have it, though. That's the difference. Can aerotolerant anaerobes grow without oxygen? Yeah, there's anaerobe in their name, which means they can do that. They don't need oxygen. So we see 5 distinct growth patterns when we grow these different categories in the FTM tubes. Because what these tubes do is they set up an oxygen environment at the top of the tube and a non-oxygen environment in the bottom of the tube. So each one of these, the dots represent growth patterns within those different zones. So for this first one, right, or let me put it this way, for A, which one is our obligate aerobe? 1, 2, 3, 4, or 5? Our obligate aerobe. So what do we know about them? They need oxygen, right? They have to have oxygen, and they only grow in the presence of oxygen. So that's number 1. Does everybody understand that? Why the growth is just in the oxygen zone? Because it has to have it, and it can't grow without it. So it can't grow down here, right? Because there isn't any oxygen. So what about our microaerophiles? Think about what you know about them, what they need. Where would you see their growth? In 4, right? And again, it's a gradient in these tubes. Most of the oxygen's at the top, and then it decreases as you go down. So you see they're not right at the top like the aerobes were, right? They're a little bit below. But again, they're still in the oxygen zone, right? They're not in the non-oxygen zone. So number 4. What about our obligate anaerobes? Where would they grow? 2, right? At the bottom where there's no oxygen, right? Because they can't grow in the oxygen zone, it will kill them. Where would be, so we're down to 3 and 5, right? Do you guys see the difference in the growth patterns in 3 and 5? You see how there's more growth up here, right? Where it's uniform growth for 5. So which one do you think is the aerotolerant anaerobe? 5. Why? Yeah, they're anaerobes, right? Remember, they're not using oxygen. So it doesn't matter that oxygen is here or not here. Their growth rate isn't going to be affected. Unlike the last tube is clearly our facultative. And the reason why we know that is because we'll see an increase in growth in the oxygen zone. But we'll also see growth in the anaerobic zone, right? The no oxygen zone. So as you can imagine in the lab, what type of environment do we have in the lab? Or in this room for that matter? It's aerobic. So who is going to grow in our lab or in this classroom? Obligate aerobes and 1 more. Or 2 more, excuse me. 2 more. Who's going to grow? Will 2? No, the oxygen's going to kill it. Can we grow 3? Yeah, and it's going to grow really well, right? Because it's oxygen. Can we grow 4? In this room, Too much oxygen in this room, y'all. This is atmospheric levels. Too much. We'd kill them. Can we grow an aerotolerant anaerobe? Yeah, because, again, they can tolerate it. So as you can imagine, in lab, we only work with these three. Aerobes, facultative anaerobes, and aerotolerant anaerobes, which actually I don't have any of these right now. For 4, it's hard when you're transferring them from tube to tube because there's too much oxygen in this environment. Even that short period of time for some of them, it'll kill them. It's really hard to keep these guys happy. Which 3 are you doing now? Obligate aerobes, facultative anaerobes, and we could do aerotolerant anaerobes, but we don't have any right now. The one that we had was pathogenic, so I had to get rid of it. I got to find a new one. So like I said, Lance asked earlier, E. coli, a lot of the ones that are in your gut and stuff like that are facultative anaerobes. So why is it? Remember, it all comes down to what type of protection do they have as it relates to oxygen growth. And so for those microaerophiles, it's because they have superoctide demutase, but their levels of catalase are low or non-existent most of the time. So they can't detoxify those toxic forms of oxygen as efficiently and as well as, say, an aerotolerant like us, right? So notice we have plenty of protection. So do facultative anaerobes because they grow in the presence of oxygen. Aerotolerant anaerobes, again, they tolerate it because they have the first enzyme. They don't really need the second enzyme because, again, they're not producing a lot because they're not using oxygen. They're just exposed to it. Where those that use oxygen like us in the facultatives, we produce toxic forms in addition to being exposed to the natural ones in the atmosphere. So we have to have more protection. And then the problem for poor old strict or obligate anaerobes is they have no protection. Hence why it kills them. Obligate anaerobes or obligate aerobes. What are you saying? Number 1, if you cut out oxygen, you will kill them, yes. And if you turn it back on? No, if you kill them, they're dead. Remember, spontaneous generation doesn't happen. All right, so the problem for these guys, right, why you see no growth, right, and this is the only one, right, where you see no growth in this zone is because no protection. So what do these enzymes do? So they take toxic forms like superoxide radicals and they can combine them with hydrogen ions, protons, and turn it into a safe form of oxygen, but they still produce a bad form, which is hydrogen peroxide. But that's where catalase comes into play. Catalase can convert hydrogen peroxide into water in safe gaseous oxygen. So this is why when you put hydrogen peroxide on a wound, it bubbles. There's 2 reasons why it bubbles. One is because you're an aerobe, right? So do you have catalase? Yeah. So your cells have this enzyme so they can break down hydrogen peroxide. So when you put hydrogen peroxide on a wound, it should always, always bubble because you are catalase positive. Sometimes it won't bubble. Well, that's because you just basically poured mostly water on yourself. Because over time it will break down into water and gaseous oxygen. This is why sometimes when you open the hydrogen peroxide bottle, it kind of makes a fizzing sound. That's the oxygen being released. That's a bad sound when it comes to your hydrogen peroxide. It means it's too old and you probably need to throw it out. There's an old wise tale out there that says that if you pour hydrogen peroxide on your wound and it doesn't bubble, it's not infected. That's bullshit, right? Because I just told you it should always bubble because you are what? Catalase positive, right? Now some microbes too, right? They have catalase if they're aerobes. That bubbling action is really good when you're trying to clean stuff other places, right? You can use hydrogen peroxide anywhere else you want. You can use it as a mouthwash, but I'll tell you it's disgusting. They add it to your mouthwashes nowadays and your toothpaste and stuff like that. The bubbling action helps. And basically when you put it on your wounds, you were basically just throwing Katrina on them, like more than they could handle, right? The problem is it causes destruction to even your own cells. That's why they suggest you don't use it anymore. And you certainly would never want to take it internally, right? It does tell you not to do that.
I do that often on wounds.
Yeah, skip it. Soap and water, right? Triple antibiotic ointment. That's your best course of action. If you want to clean your jewelry, right, and you see that bubbling, you definitely know you got a whole bunch of bacteria crud, and the bubbling helps break all that crud off, like say your earrings or something like that, right? You can use it to clean things in your house. Just don't use it to clean your body anymore.
So, I mean, it's still on shelf.
It's still on shelves. You can still use it for other things. It's just recommended you don't use it on your skin anymore, on a cut on your skin. Creates more damage than it does help. Skip it. Yeah, I know. Okay, so a microbiologist that wants to work with anaerobes has a problem, right? Because we need oxygen in the lab, otherwise we're going to pass out and die. But we need to provide the environment necessary for them to grow. So some labs that work with a lot of anaerobes actually have a machine that has a door where you put it in here, and then there's another door in here, and you work in an anaerobic environment through these gloves. I had a colleague of mine that worked in a lab, and this thing kept springing a leak, so her cultures kept dying, right? Because if oxygen gets in, it kills them. So she gave up, and she switched to my lab, which is the immunology lab, and we didn't work with anaerobes. In other labs, you may use jars like these where you can create a reaction inside where it sucks the oxygen out. But again, you have to work with organisms that aren't really, that can tolerate like a little bit of oxygen for a short period of time before it's turned into a non-oxygen environment. And so we have some of these in the lab. Unfortunately, a lot of ours have sprung leaks. The seals don't stay well. We don't have enough for each lab section to actually do the experiment anymore, so we look at pictures and talk about the experiment we used to do. These things, unfortunately, are like $500 a piece for the containers. So yeah. So what type of equipment is very popular now that we actually use this to actually prevent what type of organism to grow? So if we're sucking out, right, so there it is, right? They're sucking out the oxygen from the container, or in this case, the food saver bag. We're creating what type of environment?
I saw that at Target.
Yeah? We're creating an anaerobic environment, are we not? So who are we killing if it was in there? Aerobes, microaerophiles. Who are we not killing? Anaerobes. Who else? Let's look at our possibilities. Right? No oxygen. So we're killing obligate aerobes, and we're killing microaerophiles. Are we killing strict anaerobes? No, we just made them happy, didn't we? Are we killing aerotolerant anaerobes? No, they're anaerobes. Are we killing facultative anaerobes? No, they're anaerobes. We just made them really happy. Mm-hmm.
So, can they like be like microbes that spoil food or something?
Right, we're gonna get to it. Does this make sense to you guys? So this is why too we usually don't use just one method when dealing with trying to preserve our food, because this one just eliminates aerobes and microaerobes. You just made it a really favorable environment for anaerobes. So a lot of times we suck the air out and we throw it in the freezer, right? We use 2 levels of control, temperature and the removal of oxygen. So the next that we use a lot too in our preservation of our food is the different variations in pH. So pH is the measure of hydrogen ion concentration within an environment. And so there are some organisms, as it implies, can live in an environment where outside of that cell it's acidic. Those are called acidophiles. Others, the outside environment has to be pretty much in a neutral range for them to survive. Those are your neutrophiles. That's what we are. Right? If our environment isn't neutral, for like our blood cells, it's not good. Alkalinophiles can survive in environments where there's an alkaline or basic pH outside. But for the most part, all organisms maintain a neutral pH range within the cell. So this is talking about what kind of environment can they live in. So what happens if they can't maintain a neutral pH inside? They'll die. So we use this in controlling microbial growth, right? A lot of the cleaners that you use create pH imbalances. A lot of the chemical cleaners you use are either acids or bases. You're trying to knock them out of their comfort zone. So these numbers, unfortunately, you do need to know. Acidophiles are ones that can grow at 5.5 or lower pH. Neutrophiles are ones that can grow an external pH of 5.5 to 8.5. And alkalinophiles can grow at a pH greater than 8.5. But again, this is the external environment, not inside the cell. Inside, they've got to maintain relative neutral pHs, otherwise they're going to die. But some of them, of course, have adapted to be able to live in acidic environments. So if you throw an acid at them, is that going to kill them? No. But if you throw a base at them, right, that would kill them. Your alkalinophiles, if you throw an acid at them, they're not going to be able to resist that. That's going to kill them. Where bases probably aren't going to bother them, things like bleach. But vinegar would take care of an alkalinophile. Neutrophiles, we're lucky. You go acid or base and they're not going to be happy, right? Either way, they're going to be disrupted and harmed by that. The next is temperature. So there are some organisms, again, that can live at different degrees of temperatures. And we have 5 different classifications for these. You need to be able to list them in order and have a general understanding of the environment. For these, you don't have to memorize the temperature ranges. Thank goodness, right? Because they give them in Celsius, which is even more confusing. So we use this all the time for controlling growth, right? We use temperature all the time, whether to slow them down or to kill them. There's one form that she was alluding to earlier, someone was alluding to earlier, that really creates some problems for us. And that's endospores, right? Because endospores are a dormant form of that cell, right? It's not actively metabolizing them. So, changing pH isn't going to really affect them. In forming the endospore, they dry themselves out, so removing water isn't going to be an issue. That protein coat that it forms around the DNA to protect it is pretty stable. So most forms of heat, even all the way up to boiling, is not going to disrupt it. UV light isn't very penetrating, so it's not going to get through. Even some forms of radiation can't get through. Whoa. That's frightening, huh? So some organisms, right, that are aerobes, that are strict anaerobes, they can form endospores. When oxygen level starts increasing on them, they're like, oh crap, I gotta go into hiding. But they're resting, right? Then you stick them, you suck the air out of that bag, right? And you just stuck them in an anaerobic environment. Those spores go, yay! Party time, right? Uh-oh. So now they start growing. Right? They bust out of that spore form. They go back to the vegetative form. They start metabolizing and growing. And there's one that we have to worry about in our food because it can do funky things like this when we do things like canning and sucking the air out, right, is Clostridium botulism. Because it is a spore former, but it's an anaerobe. So most of the time, if there's oxygen, it's not going to grow. But if there's spores in there and you suck the air out, you just made it favorable for them to grow. So most organisms, right, fit into one of these classifications. They really can't go too hot or too cold as it relates to their classification. And you'll see archaea are our extremophiles, right? They can pretty much, any niche as far as temperature, they can handle it. But we're not talking about a single organism, we're talking about that whole group of organisms. So some of them are living up here, others are here, right? Other organisms are here. We're mesophiles, right? You can't go too hot, you're going to melt us, denature our proteins, our DNA, melt our membranes, we're going to die. And we really can't survive in really cold temperatures, right? Things don't move for us. So in Yellowstone National Park, these pretty colors that you see, these are microorganisms living in this hot spring that actually, different ones produce different colors in the different temperature ranges that you see here. Right? That's why they get these beautiful colors. So in order, the ones that live in the coldest environment are your psychophiles. Psycho actually means cold, and as far as I'm concerned, if you love cold, you are crazy. Then comes our psychotrophs, and then us, right, the mesophiles right there in the middle, then thermophiles, then hyperthermophiles. So what type of ranges are we talking about here? And again, this is in Celsius scale, based on the properties of water. So water freezes at 0 degrees Celsius. It boils at 100 degrees Celsius, which is 212 degrees Fahrenheit. Body temperature is around 37 degrees Celsius. That's why one of our incubators in the lab is set at 37. So these are just some references to help you because you're probably not used to seeing Celsius. So psychophiles are living in glacial lakes, right? Just at about almost freezing, right? The colors that you see in the icebergs and things like that and the glaciers are from organisms growing right before it froze, and then they're suspended in that ice. So those pretty colors that you see are microorganisms that grew when it was in its liquid phase. All organisms need water in its liquid phase. But they kind of can go into a state of animation where they just hang out. Bacteria you stick in the freezer, y'all, you don't usually kill them unless ice crystals poke them. You just put them in a dormant state. When you take it out of the freezer and you defrost it, they start growing again. This is why you don't want to do this repeatedly. Psychotrophs are the ones we worry about because this temperature range is about your refrigerator. And there are some microorganisms like Listeria that can grow in the refrigerator. Mesophiles is human body temperature. So these are all the pathogens that grow inside of us, right, are mesophiles. Thermophiles are things that you would see in those hot springs and a compost heap. This is really high temperature. This is 158 degrees Fahrenheit. That's hot. We don't really have to worry about these guys being pathogenic for us because we're too cold. And hyperthermophiles, these are in hydrothermal vents where volcanoes are erupting under the ocean. They've actually found microorganisms that can grow at that very high temperature. And they don't melt and fall apart. So how do we use this? Well, in the lab, which actually is in the biology lab, we use heat to sterilize, but we use a special form of heat. Under pressure, moist heat, it's actually steam, in a machine called an autoclave. And that is the only way you can actually destroy endospores, is under pressure with moist heat steam. So they use something similar to this in the canning process as well to be able to kill the Clostridium botulism endospores, to make sure they don't get in the cans and they don't grow. Yeah. So what you do at home isn't enough because it's not under pressure. They do it under pressure in the commercial canning industry. That's enough to disrupt the endospores so they can't germinate in those anaerobic environments of the can. So but otherwise, for the most part, if it isn't an endospore, right? Most of the organisms that would make us sick, right, those mesophiles, we can heat. That's why we cook our food, y'all, is actually to destroy the microorganisms present. And that's why you need different temperatures for different meats, because of the possibility of different organisms in those meats. What do we use refrigeration for? To slow them down. What do we do freezing? To basically stop them, put them in suspended animation. It does not kill them, though, for the most part. It may kill some of them, but not all of them. Long-term storage in the microbiology lab, all my stock cultures are in my freezer. Right? I can give them some food, warm them up, and they're like, woohoo, yay, happy. What do you want me to do? Yeah, in the lab, not at home. Well, at home we all have it too, right? We just don't usually want to cultivate them. All right, so the last is water activity. And so for this one, again, all organisms have got to have water. And some of them can resist not having good levels of water. We refer to those osmotolerant. Others have adapted so much so, like your halophiles, that if you suck the water out, they, for the most part, can still survive. So we use hypertonic solutions. What type of hypertonic solutions do we use to preserve our food? What can we add to a solution that's going to cause the cells to lose water? Salt. Salt is the most common, right? And so that stresses them by causing them to lose water. But they can bounce back from this, y'all. That doesn't kill them. So again, we usually use multiple methods, right, when we're trying to, so curing meats and stuff like that, right, using salt and stuff helps, but there are some organisms that are resistant to this. Some that actually pervert this environment, referred to as halophiles. Pressure, as it implies, right, we use pressure to crush them and kill them. Ones that can resist that are referred to as barophiles. We've already learned about radiation, right? UV light.
Did you say barophile?
Yeah, barophiles. Barophile. For radiation, we know that UV light can damage DNA, right? What does it cause? Thymine dimers, right? So we can use that to cause mutations. X-rays and gamma rays can literally chop up DNA, right? And if you don't repair it, that can cause cell death as well. All right, so there's our thymine dimers. This is, I'm going to decrease part of this objective for your guys' test. So I am not going to have you know all the properties of these three media. But you do need to be able to define what we mean by synthetic versus complex medias, selective and differential, and the fact that the media, the food, can come in two forms, whether it be liquid or solid. What makes it solid? So why is oh, come on, Erica. You can do this. I hit it. Where'd my webcam go? I wanted it back. I hit it good. Oh well. I have no idea where it went.